The action potential arrives at the synaptic knob, opening up the calcium channels. Once the calcium enters the membrane, it causes the vesicles filled with acetylcholine to fuse with the membrane. The acetylcholine is released from the synaptic knob into the synaptic cleft. The acetylcholines bind to the acetylcholine receptor, opening up the channels that allow the sodium to enter into the motor end plate of the skeletal fiber. The increase of ions inside the muscle cell creates an action potential in the sarcolemma.
Once the action potential enters the muscle, it runs into groups of fascicles that are made up of several different myofibers or muscle cells. The myofibers are surrounded by the sarcolemma, which is represented by the green zip ties. Each myofiber is made up of a myofibril. Once the action potential hits the sarcolemma or cell membrane of a muscle cell, it travels down the T-tubules, which go around the myofibrils, and hits the sarcoplasmic reticulum, which contains the calsequestrin and extra calcium, which is then released into the sarcomere, which is made up of several different parts. Inside the sarcomere, we have the A band, which is the darker area consisting of both thick and thin filaments, the I band, which is the light area consisting of only thin filaments that also includes the Z disc, which separates each part of the sarcomere. The H band is the lighter area inside of the A band, which is made up of just thick filaments, and the M line suspends the A bands in space and allows for the sarcomere's unique appearance.
The calcium floods over the thin filament and it binds to the troponin, causing it to release the tropomyosin from the actin active site. The high energy myosin heads leap up and bind to the active site on the thin filament, forming a cross bridge. The myosin head pivots to the low energy state, releasing the ADP and moving the thin filament over the thick filament. This is called the power stroke. ATP binds to the myosin head, causing them to break free from the active site on the actin. The ATP, which was produced in the mitochondria, breaks down to ADP, which powers the myosin heads and puts them back in their powered state. The ATP is also used to pump the calcium back into the sarcoplasmic reticulum, which releases the troponin, which causes the tropomyosin to cover the active site on the actin once again.
This, along with the breakdown of the acetylcholine by the acetylcholinesterase and the synaptic cleft, signals the end of the muscle contraction.